good afternoon, everyone. My name is Javier Cubero. So welcome to the Young Investigator seminars of today. In today's seminar, we have with us Olga Estévez Vázquez. She belongs to the Department of Immunology here at the School of Medicine in Madrid, at the Complutense University. And she's working with Yulia Nevzorova in her PhD thesis. So she did a short-term scientific mission in Turkey with Özlem, who is also here, I think as well. And she will show you very nice data on DIli and Zebrafish. So we are waiting for your presentation, Olga. So whenever you want, you can start.
Thanks, Javier, for introducing me. I will share my screen. Just a moment.
Hi,
Can you
everyone.
see? Hi, Özlem. Nice to see you there.
Hi, Olga.
Hi.
Hi, Özlem.
Hi, Javier. Hi, everyone.
If Özlem wants to say something about Olga and the short sensitive mission she did, you are most welcome.
Yeah, thank you. I just will listen to her. It was a great experience, really. Thanks.
Can you see the screen correctly?
Yes, yes.
Okay, perfect. So thanks, Javier, for introducing me. As Javier has said, today I'm going to share with you some of the results that I have got from my STSM stay in Turkey. The project was based on drug-induced liver injury in the context of dual modeling Zebrafish. So first of all, I want to put into the map the places that I have been into. As Javier has said, I'm actually a PhD student in the immunology department of the Complutense University in Madrid, and I have traveled to Ankara, capital city of Turkey, to stay in Özlem Konu's lab in Bilkent University. And finally, during the last weeks of my stay, I have gone to Izmir. There I've been into IBG, Izmir Biomedicine and Genome Center, where I have finished my experiments on Turkey. Total amount of time that I have been there, it was 3 months. So first of all, I want to talk a bit about NAFLD, or non-alcoholic fatty liver disease. It's probably something that you already know, but I want to refresh your mind. So what is NAFLD? NAFLD is the leading cause of liver disease in the Western world. It's an umbrella term, and it englobes from earlier stages of the disease, which is just steatosis, which is fat accumulation into the liver parenchyma. And this disease, it can progress into more advanced stages of the disease, which is steatohepatitis. If the disease continues to progress, it can develop fibrosis and even end into hepatocellular carcinoma. And what is DIli? DIli, or drug-induced liver injury, it's a liver disorder caused by drugs or other non-pharmacological compounds. It is known that some drugs can aggravate pre-existing fatty liver and also accelerate the progression of the disease into more advanced stages, as this steatohepatitis that I have just mentioned. So it is known that there is a reciprocal effect in between NAFLD and DIli. So the drugs can cause NAFLD by acting as steatogenic factors, and pre-existing NAFLD could be a predisposing condition for certain drugs to cause DIli. What is the result of this positive retroalimentation loop? It can generate an induction of hepatic oxidative stress, also an impairment of mitochondrial fatty beta oxidation, and an increase in the uptake of fatty acids. So for our studies in Turkey, we have decided to use as an animal model the zebrafish. Maybe some of you are not that familiar with this animal model, as I was not until that point. So I want to share a little bit of information about zebrafish and what are the advantages of using this animal model compared to, for example, using a mice. So it is known that the zebrafish is really similar to human in the hepatic cellular composition, function, signaling, or response to injury, and cellular processes that mediate the liver disease. Also, the lipid metabolism is really comparable to humans. They are much cheaper, and you can use a bigger sample size. The development of the embryos is much faster, and you have liver and digestive organs that are already mature at 5 days post-fertilization. They are also easy genetically modified, and the optical transparency of the larvae enables real-time monitoring. So how do you create new embryos? You need to set up some breeding tanks. And how you set up these breeding tanks in the zebrafish? Here you can see a picture from the Bilken University Zebrafish facility. And what you need to take is at least 2 females and 1 male and put them into these special tanks that they are prepared for embryo delivery. The fish will be separated overnight during this with this plastic separator, and in the morning you will release the separator, and a few hours later the embryos will be delivered into the water. So you can see you can just set up the breeding tanks one day, the following days you already got your embryos in the morning. So it's true that the zebrafish life cycle is quite fast. You just start with one cell that starts to divide 30 minutes after fertilization. And 6 hours later, the gastrulation or the division of the layers that will origin the organs in the body of the fish are going to start to develop. Also, just 1 day after the fertilization, the major organs of the embryos are already developed. And 2 days post-fertilization, the larvae, they are already able to free swimming. So how do they look like, these embryos? You can see here, this is a Petri dish, and these white dots, they are just the 1 DPF embryos. If you put the plate under the microscope, you can perfectly distinguish the fertilized and alive ones, which are here in the right, and also the dead ones that you need to remove. So if you put it also under the microscope, 1 by 1, you can distinguish perfectly just 1 day after the fertilization all the parts that I have been talking about. You can distinguish the chorion to protect the embryo, and also this big yolk sac, which provides the proteins and all the nutrients that the embryo needs to survive during the first days of their life. So to set up these breeding tanks, it's also you also need to distinguish in between females and male zebrafish, which can be somehow tricky at the beginning. The difference mainly in between the two sexes is the females, they used to be a bit bigger, and also the belly used to be a bit thicker, especially if they are full of eggs. The pattern of colors is somehow a bit different, but this is a bit more difficult to distinguish, especially in certain individuals. Also, there are a lot of strains of zebrafish. One of the ones that we have used, and they are the ones most used, is the wild type AB zebrafish line, and also the Casper one. The Casper has a really big advantage. These fish, they are completely transparent, so you can really do a good monitoring just by the outside of the body. And how is the dissection? This was somehow interesting from me because I'm used to work with mice. So here everything becomes like more tricky, more small. And you need to put your fish inside a tank with water and some ice, and you can add some trichine as an aesthetic to make the fish dead. After that, you can start with your dissection. What can you find when you open your adult zebrafish? You can find all the organs easily visible. Here you can distinguish the liver, that is somehow mucus texture, and next to it you can find the intestine. These were the organs that we were more interested to check for our dual and DIli test. So another interesting thing to take into account is that the architecture of the liver, if you compare the mammals and the zebrafish, is quite different. In the mammals, the liver is perfectly organized in liver lobules. These liver lobules, it contains the portal trials on the corner, which are formed by bile duct, the portal vein, and the hepatic artery. The blood flows through the portal vein and the hepatic artery through the sinusoid into the central vein, and the hepatocytes, they are disposed into the main cells. However, in the liver parenchyma of the zebrafish, everything is completely located in a different position through this parenchyma. And here you can see a really nice picture. The liver will be in this area of the fish, and here you can see an hematoxylin eosin staining of this liver in the adult fish. So the main aims of our project was to establish a dual plus DIli model in laboratory zebrafish, the Dario larvae, and also to characterize the phenotype of the dual plus DIli experimental zebrafish model with a particular focus on the process occurring in the liver and the intestine. Everything was based on a paper that we have published in our lab, which was called an experimental dual model of advanced liver damage. We have developed this dual model in adult wild-type mice, and we have combined it Western diet plus glucose plus ethanol, and with it we want to mimic what is going on in the individuals that has intermediate alcohol drinking pattern and also signs of metabolic risks, and you can finally see a lot of these patients in the daily clinical of a doctor. So how we divided our groups in this dual model? We want to perform this dual model in both adult and in larvae zebrafish, and for it we have established 4 experimental groups. In one side we started with a control with a commercial diet. Also we have an ethanol control group, which is control diet plus ethanol 0.25%, and we have also set up 2 other groups, one with Western diet and other with this dual diet, which was the combination of the Western diet plus ethanol 0.25%. So you may think, what is the difference in between the control and the Western diet? So the main difference in between the composition of these 2 diets is the content of palm oil, cholesterol, fructose, and fat. That is much higher in the Western diet compared to the control ones. We have commercially acquired this zebrafish feeding, and it's important to take into account, if you are going to develop the model in larvae and in adult, the size of the pellets for larvae should be much smaller than for adults. So first of all, I will start showing with you the larvae results that we have got. The first part of our experiment was to set up this certain ethanol concentration that will be more suitable for developing our model. For that, we have selected A, B, and Casper strain and 5-DPF larvae. We have started with 0.25% ethanol concentration and increased it into 1.5 and 2%. But we have realized that really high concentration of ethanol generate a big debt in the amount of larvae that we are having in each pet dish. So we decided to lower a bit the amount of ethanol that we were giving to the larvae. So for that, we have used again AB and Casper 5-DPF larvae, and we set up these experimental groups, control and ethanol 1%, and then Western diet and dual diet. We have set up 2 different concentrations of ethanol to check how was going on the behavior in this larvae. We also use this fructose 2% larvae as a positive control because it is known that fructose increase the amount of lipids that they are accumulated into the body of the fish. First of all, we need to check our larvae and select them under the microscope, and we have put an amount of approximately in between 40 and 50 larvae per well. And we have been doing daily control of the larvae by taking pictures under the microscope. Here you can see the 7-DPF larvae, which is 48 hours after we have added the treatment into the Itris media. So we can start to see that some of the larvae, especially the dual 1.5, they are starting to have some changes in the morphology. If you compare it with the control ones, they are starting to become a bit curvy and also a bit shorter. We have also done the same experiment in Casper fish. Here the pictures, maybe it's a bit more difficult to distinguish them because, as I told you, the Casper line is completely transparent. But again, we can see that some of the larvae, they are starting to lose the shape of their body. One day later, when the larvae are 8-DPF, so 72 hours after the exposition to the treatment, we can see that the tendency is again the same. So even in the ethanol group, they are starting to become a bit curvy. We can also see here, especially in this image, is really clear that the larvae, they are eating the food that we are putting into the Itris media. The same was going on into the Casper fish. So at that point, we wanted to check what was going on with the lipid deposition in the inside of the body. So we performed Nile red in vivo staining at 8-DPF. So the Nile red it's stained in red, the lipid accumulated inside the liver, the body of the larvae. And as you can see here in these pictures, the larvae treated with the Western diet, dual 1% and 1.5%, they have an increase in the accumulation of fat inside the body. I will explain you a bit here. This line, it will be the lipids accumulated into the intestine, and this area over there that is more enlarged, it will be the liver area. So we can perfectly distinguish that, especially in this Western diet and dual 1.5, the liver area is completely red, much more compared to the control one. It means that there is a significant increase in the lipid accumulation in the dual and Western diet groups compared to the control ones. And we did the same in the Casper group, and the pattern was exactly the same. Especially we got a really nice staining in this individual over here with the lipids accumulated in the liver and intestine area is really huge. So we continue for one more day, and when the larvae were 9 dpf, so 120 hours after the exposition to the treatment, we see that the shape of the larvae, it's even becoming more weird than at the beginning. Some of them, they are really curvy, so they look really unhealthy. And moreover, we only not just find that the shape is changing, we find that there is a lot of dead fish. Even in some group, we see that after 4 days of exposition to the treatment, the dead were starting to die in a big percentage. So at that moment, we wanted to combine our dual model with the APAP treatment. For that, we have used a 5 dpf larvae, AB and FAPB strain, and we have set up the same experimental groups as before. So control, ethanol, Western diet, and dual, but we have add APAP 0.25 millimolar. This is APAP, it's paracetamol. So we have added into the E3 media. And let's see what has happened in this case. So here you can see on the right, the 4 groups with APAP, and on the right, the groups with APAP. And I have overlaid the bright field image with the fluorescence one. We have performed again the night red in vivo staining at 9 dpf, and we can see that we have a really nice stain, so a big lipid accumulation in all the experimental groups, but especially we compare with the ones treated with APAP. It is true that there is a high increase in the lipid accumulation in ethanol, Western diet, and dual group compared to the corresponding control ones. We also performed the same experiment in the FABP strain line. This strain has really a good characteristic. They are genetically modified and they have fluorescent heart in green and liver in red. The problem here is that we didn't get that really good images. Still, in the dual group, we can see that these individuals, this will be the heart. It has autofluorescence in green, and this will be the liver, autofluorescence in red. And we can see that this liver, especially the one treated with APAP on the right, they are really big to compare to the control ones. It's really a pity. We need to repeat this experiment because the images here, they are not that good. And at the end, after doing the in vivo part, we want to do embedding of the larvae, but we didn't have that much time to improve the protocol. Still, I want to show with you this image because it's somehow cute how they are so small located in this agar mold. From that point on, we decided to collect and make a pool of these larvae and check the gene expression on them. So for that, based on bibliography, we have selected 4 genes that were already studied in dual mice and zebrafish, Western diet and ALD models, and designed primers for zebrafish liver and intestine samples. So we have selected 4 key genes involved in 4 different cell pathways. From lipogenesis, we have chosen SRBF1, from beta oxidation CPT1AA, from ER stress DDI3, and from inflammation TNF-alpha. And for that, we have collected 10 dpf Casper zebrafish larvae, and by doing a pool of them, we have checked we did RNA extraction and checked by qPCR the expression of the CPT1, SRBF, and DDI3. And we can see that there is a slight increase in all the treated groups if we compare it to the control ones in the 3 genes that we have studied. So it looks like that there is a slight tendency in the increase of beta oxidation, lipogenesis, and IR stress markers in this Casper 10 dpf larvae. So from that point on, we were also working in parallel with the adult zebrafish. And what we did in the adult, we also set up the dual model using A/B strain. For that, we have located 4 aquariums, and in each aquarium, we have included 6 males and 6 females. The experimental groups were again the same. So we have an aquarium of control group, another of ethanol 0.25, other with Western diet, and dual diet 0.25% of ethanol. And how we perform this experiment, we try to mimic the same procedure that we were doing in the mice. So first of all, we try to adapt the fish to the new aquarium 4 days before starting the treatment. 2 days before starting the treatment, we change the regular diet from the fish lab to control food, the one that we have bought, the commercial one. And then at day 0, we changed in the corresponding groups the control diet for the Western diet. 2 days after, we added the ethanol to the corresponding groups. And we did the treatment during 21 days. Before sacrificing the fish, we did 6-hour fasting. But at that point, we have already found a really big problem. The fish that were treated with ethanol, they were starting to die. So at the end point, we have collected the fish and do a biopsy of all of them. We have measured these parameters. On one side, the body weight and body length. We see that there is a slight tendency in the treated groups with ethanol, Western diet, and dual. The body weight of these fish is slightly increased compared to the control groups. However, we didn't find significant differences even in body length or in the body mass index. We also collect the liver and measure the size of it. And also we have measured the length of the intestine. But here we also didn't find any significant differences. We also check the basal glucose by using a glucometer, cutting the tail and taking a little drop from the fish. And we also didn't find any significant differences at that point. However, we collect the fish and we did some embedding and staining. And at that point, some differences started to appear. So here you can see the pictures of H&E staining of liver, male on the top part and female on the bottom part. It is important to note that the liver architecture in the male compared to female zebrafish is quite different. So in the control group, the males, they already have more steatosis in the liver parenchyma than the females. But if we go on into the experimental groups, we can see that the treatment, ethanol, Western diet, and especially dual diet, they generate an increase in the steatosis in the liver. You can see here in this dual picture that there is a big amount of lipid droplets inside the lipid parenchyma. We can distinguish even microsteatosis even inside the nuclei, which is a characteristic from zebrafish. We don't find these intranuclear lipid droplets in the mice, and we can also see some macro lipid vesicular lipids surrounding the liver parenchyma. Sorry. And moreover, we also collect the liver and the intestine of this zebrafish and check the gene expression as we did before with the larvae. So we have checked the expression of CPT1, SRBF, DIli, and TNF-alpha in the liver of this zebrafish. And we didn't see any significant differences in any of them. However, the Western diet group has a slight tendency in the increase of all these 4 genes. But if we go to the intestine expression of these genes, we can see that in the dual group, the expression of beta oxidation marker like CPT1, also SRBF, DIli, and TNF, they are increased compared to the Western diet and control groups. And we have found the bigger difference in the ER stress marker, DIli, in both Western diet and dual group if we compare them with the control one. So I have made this graphical abstract as a summary of what I have been talking until now. So it is known that in patients that have NAFLD, so they are obese, they have type 2 diabetes or other metabolic disorders, and they are taking drugs, these drugs can act as a hit for the development of insulin resistance, which is an increase in the insulin in the bloodstream and also glucose. These individuals, they also have an increase in the uptake of free fatty acids into the liver. These fatty acids, they will be accumulated into triglycerides. Also, the novel lipogenesis is increased in these individuals, and SRBF, one of the genes that we were studying, it is involved in this cell pathway. Normally, beta oxidation is decreased, but we have found in our model that the CPT1 expression, it was increased. And it is known that the lipophagy, so the degradation of the lipids, it is also decreased. Apart from that, the exportation of lipids from the liver, it is decreased. All these procedures, or the alteration of these procedures, they generate inflammation, so the increase of TNF-alpha, and this generate a lot of ER stress. The ER stress, the DIli is one of the genes that is involved in this procedure. It generates the activation of the hepatic stellate, and as consequent, fibrosis start to develop. So, as conclusion, I have put here first the results that we have got from the larvae and then from adult. So I started with larvae. The dual model has been established in larvae zebrafish. We can also conclude that the larvae treated with dual diet has developed changes in their morphology and length. Moreover, the larvae fed with dual diet and APEP develop hepatomegaly. And also the lipid accumulation in liver and intestine is increased in the larvae fed with dual diet and dual place APAP. Going now into the adult part, we have got that the dual model is established in adult zebrafish. However, it needs to be improved. Also, the adult fish fed with dual diet has a tendency to obesity, and lately, dual diet generates hepatic micro and macrosteatosis. So as future aims, I have put here 3 important points. First of all, it will be good to increase the experimental number, also to obtain serum sample from the fish to be able to check the transaminases level, and to check the different marker of oxidative stress, beta oxidation, lipogenesis, and inflammation, between others, to analyze which are the pathways that are more affected by the treated administration. So, considering the growing evidence of obesity and the high prevalence of NAFLD in the population, the study and the impact of DIli requires extensive investigation. So this was just the first stone of a big pyramid that I hope we can continue at some point. So just say thank you to the hosting institutions, especially to Özlem Konu, my PI in Bilken, and to Gulçin Çakan in Izmir, and all the students, predocs, master, postdocs, technical technicians, and everyone that was helping me during this procedure. And thanks for everyone to attending to this talk. Çok teşekkürler. Thank you.
Thank you.
Thank you, Olga. Thank you for this nice presentation.
Thank you.
Okay,
Yeah.
to you, Özlem, you want to say something?
Yeah, I just want to say thank you for actually to Olga, to Yulia, to Javier, to the cost. It was an excellent experience for all of us, and Olga was excellent. She worked so hard, and it was the summertime, but it doesn't matter. She worked, and it is, as she said, that these are quite preliminary still, but I think it's very promising, and hopefully we will continue this with the EU project and with other projects, and we would be happy to have Olga back, or anyone who wants to study. Thanks a lot for this opportunity, Olga, and excellent presentation.
Thanks a lot, Özlem. Thank you so much.
Thanks.
Yeah, as you said, we will continue with this because it's the basis of one working package of the European project.
Yes.
So one of the aspects I want to emphasize is that not all NAFLD patients are obese. Yeah, there is a big problem because many NAFLD patients are normal weight. So to detect the NAFLD there is difficult, no? Because you have the gold standard, which is the biopsy, but other techniques need to be, need to be elucidated, no?
Yeah.
So, so I have some questions regarding you. So you said, Olga, that in the zebrafish is a very nice model for lipid metabolism, no? Specifically to mimic with the human samples. Did you check changes, because you show with the dual diet changes in the ER stress genes? Did you do it also with APAP?
No, the main problem here was to achieve the APAP part, you really need to set up the dual part. So APAP was really the last thing. So we didn't have time at that point to do any RNA structure or anything. So with APAP, I just did the in vivo part. But for sure, it will be interesting to do some pool of larvae and do some gene analysis. Yes, we didn't have time, but it will be good.
It was striking, no? The differences that you had with the Western diet, but not with the dual diet in the liver. Do you think this might be reflection of that the zebrafish are very sensitive to the dual diet, too sensitive, I would say, and maybe they are better for a Western diet?
For sure, they are good for a Western diet. However, what I say, the dual needs to improve because it's true that they are somehow sensitive. The larvae, somehow they are even more resistant to ethanol treatment than the adults. So maybe we need to try at some point some intermittent exposure or something that is not that damaging for the just a continuous exposure with ethanol. Maybe it's really too much for them. So
yeah, this is what I think. Yeah. It might be.
Yes, we need to check also this.
Some intermittent, but of course, in 3 months, this is the only thing you can develop. I mean, you need more time.
Yeah, exactly.
For this. Did you also check? Can you do? Well, you show H&E. Can you do serous red staining for collagen deposition?
Actually, I brought the samples here and I did it, but I didn't see. The thing is that you do the embedding of the whole fish, and the liver is really a tiny part. So I was not able to see any fibrosis in the ones that I took here. Okay. However, I just took one male and one female, so this is not representative, but I did it actually. I didn't put it, but I did it. Well,
it's very peculiar, no? The difference of the liver architecture between male and females, no? It's really.
Yes, they are really different, and also if we are doing some lipid studies, we need to be sure of which sex we are choosing, because the males, they already have somehow big amount of steatosis in control group. So this is important to take into account.
Okay. There is one question from the audience, Guru. I think he had the same question in Turkey. He asked Oslan the same question, I think. What is the you are there, Guru. You want to ask it by yourself, or you want me to read it?
I just saw the fluorescence organs, heart and liver, particularly liver. You said when you gave APAP in the combination, you said it was bigger. I want to know, what does this fluorescence represent, and do you quantitatively estimate it, or is just like a look? Is there a validated method? The reason I'm asking is that you can bind stuff to acylglycoprotein receptors in the liver and assess their function, not just look at the size. And with APAP, I mean, size might be inflammation, size might be just fat. So you can't really tell how APAP has added to the model.
Thank you so much for the question. It's true what you say. We can't say that much from that pictures. Here you can just see the size. You can't say much about the function or anything else. I just showed also, you see that the controls were not that good, but it was just the last experiment that we have performed. And this FABP line is really good just to check the size, because just by eye, you can see by fluorescence the size of the liver. And we wanted to see in that case if it was some hepatomegaly just by eye, putting under the microscope. But you are totally right, this should be continued step further and to check what's going on, if it is really fat accumulation or inflammation or what's going on there.
And
Yeah,
also,
you're right.
I think,
Thank you.
Olga, I think you can measure the fluorescence if you have like enough sample. You can get the intensity and like make some sort of an intensity measurement, which can tell you significance a little bit better than, you know, just by looking at it. But apart from that, like a comment to Guru is, I think liver size is used, but like you're right, it's not really indicative of or, you know, any DIli or, you know, like the loss of liver might be actually something that is like damage to the liver. You can be maybe measuring for like cell death.
Yeah.
Or if you're gonna measure like inflammation, then there are other potential strains that you can look at, immune cells, for example, the invasion of the immune cells and so on. So I think better markers are needed, that's for sure. And that will be part of the thing that we hope we can get more influence or more things to the.
I picked up on that because it allows you to investigate without sacrificing the animal or
Yeah.
chopping it up. I think so. You can really look at the natural history. So that is why I just highlighted that.
Yeah, very nice way to look at actually inside this organ, what's going on. If you have good markers of different kinds of markers, you can even have multiple, like dual or triple markers, and you can see their interactions too. So you just cross these animals and you get a dual fish, right? For the particular markers that you're interested in.
Just look at acylglycoprotein binding compound. So there is a way of measuring the function, and it gives you, there is animal model published using acylglycoprotein bound assays, which might be very good in a zebrafish where you can see everything.
Yeah, great. Yeah, look into that. Thanks.
What you can also do, I mean, with the zebrafish that I think you mentioned several times, Muslim, is just to knock out a specific gene, no? Later on. So if you find one of these genes for lipogenesis, stress, that you can go into detail and see the function, no?
Absolutely, and I think it's becoming more and more common, like not just knockout, but knock-in of specific, like human single nucleotide polymorphism or like any type of variant. And it's becoming relatively easy compared to mice. Like you just inject the fish with those constructs, and you can even look at it in F0, just in the fish that you injected. But in 3 months, you might have in 6 months, let's say, let's just be realistic, you may have a strain to have a germline mutation, if you know, or conditional temperature sensitive knock-ins. And there is, I mean, this field is coming up really progressing pretty fast.
Yes, I think, but to see the potential, you need to work with it. we have Olga here to transmit it, but I think you need to have the hands on the fish.
I think so. I mean, she hasn't worked with fish before, but I think having a like in vivo experience with mouse was really helpful. So she could really plan and do things. But of course, it's in vivo. You don't expect what's going to happen. Things happen out of expectation all the time. So a little bit more time is needed, but I think it's a very good start.
Absolutely, absolutely. Any other questions? Oh, Michelle, hi.
Hi, Javier. Olga, very nice talk. I enjoyed it very much. Actually, I would come back to one of the comments that I already made, is the difference between the pathology of the female and the male fish. Might this actually indicate that the normal chow would not be appropriate? I mean, maybe Özlem can indicate that, or you. I mean, would you expect that the livers will be different between the female and the male? Or is the chow just not appropriate?
Thank you so much for the question. Maybe Oslen want to add something else. One thing that we have in mind, it was to check, because this was a controlled diet that we have bought. So the company it has designed it accurate as control for our Western diet. But another control diet can be the regular feeding that they are giving in the fish facility. So from previous H&E stainings that they have from regular diet feeding, we have checked that the males, even with the dry regular food, they are developing this steatosis. So it shouldn't be a problem of our control food. It's something that already they develop with the regular diet that they are giving in the fish facility. Because at the beginning, I was also surprised that just the regular one was given this. Yes.
Oh, if I may go back to your presentation, you showed one slide, the differences between the female and the male, and then you showed the next slide some markers. Can you go back to those, please?
Sure. This one maybe?
Now, before that one,
before.
you see no, that one, yes. So you see here in that particular slide the differences between the male and female, right?
Yes.
So the markers you present in the next slide, are those mixed female male, or is it just female or just male?
Okay, thank you for the question. This is a good one, actually. The thing, we have started with 6 males, 6 females per group, but as I have mentioned, in both in the ethanol group and in the dual group, we have found that a lot of fish were starting to die. So we did several trials. This was due to the effect of the ethanol. So we were trying to set up different ethanol concentration, whatever. That's why I also don't show here the ethanol control group. So at the end of the experiment, from the groups with ethanol, which are ethanol control and dual, we just find like 3 or 4 fish that were alive, and we wanted also to check some H&E. So in that moment, we want to divide that 4 fish or 3 fish into RNA extraction and also H&E staining. So at the end, I just made a pool. So this is a mixture, male and female. So the thing will be to increase the N number and to do different graphs from male and from female, because you see here, for example, there are 2 that are super high, 2 they are super low. Maybe they are the males and the females. This I should check it, because you are right. It should be made a different in between different sex.
Yeah, I think so, because if it's so clear that the morphology from the liver is so different between male and female, I think you should separate. Segregate those markers and see if you can pinpoint to a specific biomarkers for the female and male. No, okay, now that makes sense. Thank you very much.
Yeah,
thanks so much.
That was a great question. I have one comment on that. The thing is, I think these preps, like these H&E stains, can also be improved probably with different fixations and times, and so it could like quantify how much more the males have steatosis than females. I think that's an important issue. It might be just a few cases, so it needs more sample size. And the experiment we did, we could only do 21 days, which was 3 weeks. We were hoping to do longer, like at least a month, or you may need a little bit more to see the effects more clearly. So all of these are planned, and I think they are absolute necessary.
No, they are very promising. The data is very promising. I think there are a lot of nice things to explore.
Yeah, the thing is, people don't generally separate males and females, and that's why this hasn't been much talked about in the literature. Is that right, Olga? We haven't seen this discrepancy. So I think this would be interesting to follow up for sure with a larger pool of males and females and separating them and analyzing them separately.
Absolutely. I mean, the morphology is so completely different, right? So I think that if indeed this is something that is okay, it's really due to the sex of the animal.
Not the chow. I think Yuchin has mentioned, right? They also see this sexual dimorphism.
Yeah, we have actually been discussing this, and then they realize with regular food from the fish lab, the males, they already have steatosis. So yes, this they already check it.
And these were all the same age, but I think the age could be another factor. Like the females, we used females pretty young, I think about still a year or younger. and they are in their fertile range. So depending on, like maybe when the females get older, this might be different. So I think all of these are interesting questions that has come up.
NAFLD actually is more prevalent in male, right? So I don't know.
It is, right? Is it? I
Is
mean,
it,
for
Javier?
some HCC, yes, but I'm not sure about the NAFLD.
Yes,
After
from HCC, I
menopause,
think it's
though.
higher in males. In
Right.
females, they are supposed to be a little bit protected with the paper from Science in the, yeah.
So maybe I can comment. So in dual mice, we had stronger steatosis and steatohepatitis in male mice. So we have steatohepatitis in both in males and females, but in male mice it was stronger.
So this is very supporting, then. It's very
Right,
interesting.
right.
Yes.
Yeah, very interesting. Okay, great. Thanks, Olga.
Thank you so much. Thanks for the question.
Any other question? Maribel, you have any comment?
I am really impressed because we are not very familiar with the zebrafish model. So I think these are very interesting results and findings. And congratulations also for the work you are also performing. And I was
It's all Olga's work. She did an excellent job, really.
Yes, Olga did a very nice
And some
presentation.
people from my lab also helped. I think they made a really good team, international team.
Yeah, really, thank you so much to all the students that goodness, Ruja, and I mean, they really helped me because I have no experience before with zebrafish. So they explain me every single detail. So thanks so much also to all of them.
To Olga.
What I knew is that dermatologists try to use this model, the zebrafish, for getting further understanding of the diseases. So I wonder if the immunological response could also be how it is similar or different to humans, and if that could be also worth
absolutely
digging into it.
Yeah, absolutely. I think using different strains that you can visualize these responses might really help, you know, non-invasively as this things goes on over a time period. I think it would open up new areas. Yeah, I agree.
Well, the difference between the genders, I mean, between male and females, in the H&E, you had a huge immune infiltration. Well, you could see the neutrophils, no, in the liver parenchyma.
But I was checking this because at the beginning I was thinking the same, but the irrigation in the liver in the zebrafish is much more. So I think some of them, they are just basals and some erythrocytes that they are not immune cells. I think so, because I was checking other H&E from, also I was not familiar with liver architecture in the zebrafish, and it makes me some confusing at the beginning. But which have checked the TNF expression in the liver and intestine, and in the intestine looks like there is a tendency in the increase in the dual one. So for sure we need to check other markers because inflammation somehow should be increased after all this bomb combination.
Yes.
Yeah, I think that, like you mentioned, the key here is the like the type of the diet, like we drink every night, you know, I mean, it's like this continuous drinking may not be the best thing for zebrafish. We might want to do like intermittent or like a binge drinking where you insult the fish and then see what happens, or the recovery process, because the fish are extraordinarily sensitive, as Javier said, and that we may not be able to see this because of the sensitivity. Something else might be happening, the cells might be dying. And so I think a more optimal level of exposure is going to make it more, you know, close to the human.
Yeah.
One of the human models of dual diet.
Yeah, I agree. I agree with that. I mean, there are a lot of things to do with this model, actually, because nothing is very well understood, no?
I think it's very appropriate for humans. So I think more models that we can compare will provide us good leads.
Yes, yes, of course. Okay, any other comments, questions from the audience? I don't see any other comment. So from my side, I just want to remind you that that the next seminar will be in I can tell you have it here written, is in next November, the 16th of November. And will be Barbara Smolkova, who will talk about mitochondria in liver development and disease from the group of Oleg Lunov, who is also here. So we hope that you can also join us the 16th in November at the same time, 4:30 Central Eastern Time.
Mm-hmm.
Okay.
Thanks for this opportunity. I think this is great. Coast, thanks Raul and Isabel and for Javier. I think excellent opportunity.
Thanks
and
to
It's a pleasure.
these talks too. These talks
Congratulations.
are great.
yes. Thank you.
Good. Thank you. Bye.
Bye
Thanks
bye.
so
Bye-bye.
much. Bye.
Thanks,
See
Olga
you. Bye.
and
Bye.
Julia.
Thanks. Bye.
Bye.